It is a pleasure to introduce you Dr. Daniel Tornero, which is our today's speaker. Uh, Daniel has a PhD in neuroscience from the University of Castilla-La Mancha here in Spain, and has more than 20 years of experience in the field of neural stem cells. His scientific interest has been oriented towards exploration of new strategies for the study and treatment of neurological disorders, in particular ischemic stroke. He spent most of his postdoctoral time at Lund Stem Cell Center in Sweden, where he developed cell therapies based on human-induced pluripotent stem cells for ischemic stroke as well. Since 2019, Daniel is an assistant professor at the University of Barcelona and head of the Laboratory of Neural Stem Cells and Brain Damage. His research is focused on the study of functional neuronal integration in cell therapies applied to brain injuries and neurodegenerative diseases. He also participates in projects related to the development of models, both in vivo and in vitro, that help to explore neurodegenerative processes and diseases that affect neurodevelopment. And with this, I would like just to thank Daniel for his willingness to share his work with us today, and just give him the floor right away. So Daniel, please go ahead. And thank you.
Thank you very much, Miguel, for this nice introduction. And thank you to CIBERNED for allowing me to share my research today here. And I would like also to thank everyone for being here, listening to me today. So today, as Miguel was saying, I'm gonna talk about functional brain reconstruction using stem cell-based therapy to repair the brain after damage. I will, first of all, I would like to give an overview of what I'm gonna say today. This is the work developed for the last 5 years in my laboratory. Part of this was done in the Stem Cell Center in Lund. And the last 3 years I have been working here in Barcelona, where I'm now, as an assistant professor in the University of Barcelona. I first want to introduce the topic where that I'm going to talk today about, and is related with the capacity to repair that we have in the brain after a damage. I will mainly use the model of ischemic stroke as a model of brain damage, but this technology can be used for many other disorders. And I have been using many different techniques, most of them related with the functional integration of the neurons after the transplantation. And what I want to try to convince you is that we are able to repair the brain after damage, an adult brain. And after injection of these neural stem cells into the brain, we are able to get these cells connected to the neuronal network, and we are able to repair the function of these damaged areas. This image was included in a review that was published last year in Frontiers and in Cell and Developmental Biology. So during my talk, I will talk about the different techniques that I'm going to use. First of all, I would like to start introducing the kind of cells that we are using. And these cells are called long-term expandable neuroepithelial-like stem cells, or LTNS cells. And these cells are generated from a skin biopsy of a human donor. After a process of reprogramming, we are able to generate iPS cells. And then through the process that we call neuralization, we are going through different steps, embryo body formation, rosette formation, and some other kinds of rosettes, until we arrive to the LTNS cells that are neuroepithelial-like cells. So they are similar to the cells that we can find in the subventricular zone. And some of the good characteristics or features that these cells has is that they are long-term expandable. So we can grow them for a long time, we can expand them, we can maintain for a long time, for many passages, and we can freeze them and thaw them several times. After applying a protocol of differentiation, we can generate different kinds of neural differentiated cells, like, for example, neurons stained in green with tubulin in this case, and also astrocytes stained with GFAP. And something that is important is to highlight that these neurons are electrically active, so they can be stimulated electrically as the normal neurons. So this is the cells that we use in our experiments. And first, I want just to introduce the first evidence that we had about the use of these cells in regenerative medicine. That was this paper published in 2013 in the journal called Brain. And in this case, we were using this model, this ischemic stroke model that affects the cortical areas of the brain. And we were using these cells, these LTNS cells, to try to repair this damage. We demonstrate in this first paper how the animals transplanted with these cells were able to recover faster than the ones without the transplantation after suffering this stroke damage. Now I'm gonna go through like a general characterization of the cells that we are generating after transplantation at the different time points. And this is just to give you an idea of how is the transplant, how many different kinds of cells we can generate from these grafted cells, from these neural stem cells. So the first time point that we used is the 2 months time point after transplantation. And in this case, we can see how most of the cells are still immature. We have many HUD cells, neural precursors or progenitors. We have also many DCX positive cells, double cortin positive cells, but we can see already like 10 or 15% of NEUN positive cells, FOX3 positive cells that are already mature neurons that at 2 months time point are arriving to this level of maturation. Importantly, we can also detect at this time point some cells that are expressing specific markers of the cortex, like TVR1, showing also morphology of pyramidal neurons at that stage. And in the Downer image, you can see also the staining for this TVR1, that is a marker of deeper layers of the cortex. But most importantly, when we go in the time, we can see how this transplant is changing with the time, is maturing. For example, at 4 months time point after the transplantation, we can see how the number of NEUN positive cells is increased. We have many mature neurons in this case. Most of these cells are also positive for KGA, a marker for glutamatergic neurons, that are the neurons that we expect to have in the cortex in the adult brain. So most of the neurons that we generate are also glutamatergic, excitatory neurons. We also can detect still some immature neurons and also astrocytes in our transplanted cells. So we can not only generate neurons, but also astrocytes in vivo after transplantation. This is all related with the grafted cells, but we were also interested in knowing what kind of cells are inside the transplant, but are not belonging to the transplant. So trying to see how integrated is the tissue in the brain of the rat. And at this level, we were observing these EVA1 positive cells, so microglia inside the transplant that is not belonging to the transplant, is not derived from the transplant, but is present inside, so it's belonging to the, so no, they are host cells, not grafted cells, but also GFAP positive cells, so astrocytes from the host, from the rat, that is included into the transplant, in the tissue of the transplant. So if we go further to 6 months time point, we can detect how this progression is increasing. So the NEUN positive cells are in numbers is higher. We only can see a few DCX positive cells, so the number of neuroblasts is reduced. and the number of cells that are proliferating is very low. The only cells that we can detect that are K67 positive, so proliferative cells, they are not belonging to the transplant, so they are not human origin. So importantly, we can detect also many different specific markers from the cortical neurons, specific of the upper layers like brain 2 or sub-B2, but also from other layers like TVR1 or CTIP2, more typical from the deeper layers of the cortex. So all the different kinds of neurons that we can find in the cortex, we can also find them in the transplant. But not only neurons we can find, as I told you before, we can also find astrocytes at the later time points. We can see a few human origin astrocytes stained here with SC123, and specific marker for human astrocytes, and also oligodendrocytes. In this case, most of the cells that are stained with these oligodendrocyte specific markers, SOX2 in this case, are more located or are concentrated in the corpus callosum, in the deeper area of the transplant. So with this brief introduction of how is the transplant, I will focus now in the main topic of my work, that is study the integration, the functional integration of the transplanted cells. And I will start with this first paper published also in Brain 2017, where we described the cells, how the cells that we transplant are receiving information from different parts of the brain. So we wanted to study if our cells, the cells that we transplant into the rat brain, are able to receive synaptic inputs from the host brain, in this case, the rat brain. So the first thing that we did is to observe in the area of the transplants using immunoelectron microscopy, if we could detect these structures, and we confirmed that we could find structures that are very well known as synaptic contacts that are from the host to the grafted cell. The grafted cells is visible in green in this picture, and is we are able to detect this because it has GFP, and we can stain the GFP with these very dark, dense spots that you can see in the image. So we confirm the presence of synaptic contacts from the host to the grafted cell. But we wanted to explore where are these cells located. So the cells that are connected to the graft are located in different parts of the brain, and we wanted to explore where are these located, where are these cells located. So to do that, we were using a very specific tool called Ravious virus monosynaptic tracing, that is based in the Ravious virus, modified, where we are removing the G protein from this virus. That is the protein that allows the virus to cross from one synapse to the other and infect the different neurons. So when we remove the G protein and we include a reporter, in this case is M-cherry, that is, it will stain the neuron with in red. And then we put an envelope in this Ravious virus from a different kind of virus. This we call pseudotype Ravious virus, stained with M-cherry, and this is what we use for this technique. In parallel, we designed a construct that is that presents inside a reporter, that is histone GFP, the receptor for the envelope that we have included in the Ravious virus, and also the G protein of the Ravious virus. And this we included into the lentiviral vector units, and we call it lentiviral tracing vector. So what we do with this technology is to infect our cells before transplanting with the lentiviral tracing vector, including this TVA and the G protein, and then transplant the cells in our model. And sometime later, we are injecting the Ravious virus. In this way, the Ravious virus will only infect our transplanted cells, but are the ones that are transformed by the lentiviral tracing vector. And this Ravious virus will travel to one synapse across this structure because this cell is expressing the G protein. this infection with the rabies virus will not go to the next neuron, because these trace neurons will not have the G protein that is necessary for this infection. So using this technology we were using in our model, we were able to detect where are the cells in the brain of the rat that are connected to our transplant through a synapses. And in these images, you can see how we observe the trace neurons into the graph that are GFP positive. So those we call starter neurons, that are the first neurons to be infected with the rabies virus. And we can see also how some of the cells in the host brain are also stained with this GFP. So they are connected to the transplant, and because of that, they are infected with the rabies virus. And we observe these cells very close to the transplant in the cortical areas, but we also can see this in very far areas of the brain, like for example, the contralateral hemisphere in the cortex, also the claustrum of these cortical areas, that is a structure dislocated in the ventral part of the cortex. And importantly, we were also observing a huge concentration of cells infected with the rabies virus located in the thalamus. And in these images, I'm showing a specific nuclei of the thalamus where we can find these neurons that are traced with the rabies virus, so connected with our transplanted cells. And we know that this is a relay step, that the information coming from the periphery of the body, it takes to go to the cortex. So first, the information is arriving to the thalamus, to these kind of neurons, and then arriving after a second synapses to the cortex. So this is the known relay step of the information, sensitive information coming from the periphery of the body. So we conclude that this distribution of these cells connected to our transplant are similar to the ones in the normal animals. So this is happening in a specific way. These kind of synapses are formed in a specific way. But we wanted to know also if these synapses were functional, were able to send information. And to do that, we did two different kinds of studies, experiments. The first one was using the Optogenetics tools, in this case, channel rhodopsin, that, as you probably know, is able to induce an activation into the neuron when we illuminate these cells that are expressing this channel with a specific light, in this case, blue light. So if we express this channel rhodopsin in a neuron, and we illuminate this neuron or the fibers, and we record from the cells that are connected to this neuron, we will be able to observe a response in this second neuron. So what we did is to inject in our rat brain after the transplantation this channel rhodopsin into the thalamus of these animals, trying to target these cells that we could see stained with the rabies virus. And then we confirmed that these grafted cells in the cortex, recording from them, we could observe a response when we were illuminating these fibers in acute slices in these animals transplanted with the human LTNS cells. The second study that we did to confirm this activity or this information arriving to the cells is in the alive animal. We were opening a window into the head of the animal to see where the transplant is located. In this image, you can see how this we can specifically see where the transplant is located, because the GFP positive cells are visible using the proper filter. And in these cells, we put one electrode, external electrode, in this area of the graft, and we were able to stimulate the rat in a particular way, in a specific way, and we detect this response into the grafted cells. So we were able to demonstrate that these cells are receiving information from the periphery, sensitive information from the periphery. So this is more or less a summary of this first paper that we were publishing, but after closing all of this, we had another topic that we wanted to explore, and this was related with not only which cells are connecting to our transplant, but also where are our transplanted cells projecting to, and if they are forming functional synapses with other areas of the brain. And in this paper, in this PNAS paper published in 2021, last year, we were able to explore this topic. So, and the first thing that we did is to try to explore where the projections starting in our transplant are going. And to do that, in the images, you can see how is the morphology of the transplant in panel B. This is covering all the cortex. In the panel D, you can see how is the limit to the area of the damage, so to the damage, stroke damage area. And also in the C panel, you can see how many fibers are crossing through the corpus callosum to the other hemisphere. And this is where we put our attention, because we think that this is a preferential area where our cells were projecting. And what we did is to explore where these projections are finishing, are ending up. And taking this contralateral hemisphere opposite to the transplant, we could, using this immunoelectron microscopy again, we were able to detect these structures, these synapses, in this case, in the other direction. So axonal terminals arriving to this area of the cortex and contacting with host cells. So our cells were able to send the projections to the other hemisphere of the brain and connect to specific neurons located in that side of the brain. And in the same case as we did in the previous paper, we explore these synapses using the Ravious virus monosynaptic tracing. And what we did in this case is to inject this lentiviral tracing vector and the Ravious virus in the contralateral hemisphere. As you can see in the image, in the red spot, this is the area where we inject both viruses. And the Ravious virus, if the cells are connected, will be transferred to the other hemisphere. And in this image, you can see how a few cells are located in the area of the transplant, are stained with the M-cherry, so infected with the Ravious virus. And also inside the transplant, you can see a few cells. In the downer image, you can see a higher magnification of one of these cells, example of one of these cells that are from the transplant and are connecting to the other hemisphere of the rat brain and infected with the Ravious virus using this monosynaptic tracing tool. So importantly, we could see how these traced cells that are in the transplant and are connecting to the other hemisphere are also positive for a specific marker called SATB2 that is in the cortex of the normal animals, and is specific from the neurons that are connecting both hemispheres. So these transcallosal connection neurons are specifically positive for this SATB2. So not only the cells in the transplant were connecting to the hemisphere, but they were also becoming the kind of neurons that they should be if they connect to this area, to this specific area. So this was very interesting, but we wanted to explore also the capacity of the projections generated by the transplant to be able to be myelinated in this case. So the first thing that we did with the electron microscopy is to observe in different areas how the GFP positive axons were myelinated. You can see 3 different images in different parts of the brain, showing how the GFP positive axons are myelinated surrounded by myelin in the corpus callosum, in the ipsilateral cortex, and in the contralateral cortex. We were able also to observe the axonal myelination at different stages. As you can see in the downer image, the A is poorly myelinated axon. In the middle one, you can see a middle myelinated axon, and in the C panel, you can see the complete myelination of an axon originated from the transplant. And as I was telling you before, we were able to generate also a few oligodendrocytes from our transplant. And in image in your right, you can see how some of these oligodendrocytes originated from the transplant, human positive, in this case with a human mitochondria marker. We could observe how these oligodendrocytes are able to generate the myelin and this covering the axons of the rat and generating this myelinization of the host brain. So not only the axons can be myelinated, but also the oligodendrocytes coming from the transplant are able to myelinate other axons. And finally, going more for the functional perspective, we wanted to explore how our cells are contributing to the recovery of the animal. And to do that, we were using a behavioral test called cylinder test, where we put the animal in a glass cylinder or transparent cylinder, where we can count how many times the animal is touching with the both upper limbs. So this was done at 6 months time point after the transplantation. And in this case was the best behavioral test to detect the efficient recovery of the animals after the transplantation. If you observe this graph, you will see how after the damage, there was preferential use of the ipsilateral side of the animal because the contralateral side is damaged, is affected by some kind of paralysis. So this preferential use of one of the limbs is completely removed or abolished by the transplantation of cells that you can see in the right side of the graph, how the use of both limbs is 50%, so it's the same. And we use this model to try to see how is the contribution of the functional activity of our cells to this functional recovery that we could observe. And to do that, in this case, we use another tool, optogenetic tool, in this case, allorodopsin, that is a channel that when you express this channel into a cell is able to completely abolish the activity, remove completely the activity of the neuron that is with this channel, as you can see in the image there. So what we did is to introduce our cells infected with expressing this allorodopsin, and in the brain after this we install optical fiber to illuminate the cortical area where the transplant is located. As you can see in the image, this rat is having one of these optical fibers that is connected, this cannula is connected to an optical fiber to illuminate the area of the transplant. When we did this experiment and we silent completely the activity of the transplant, we could not see a difference in the use of one or the other side of the body, as you can see in the image. But what it was very interesting is that in all the cases, in all the animals, the number of touches with both limbs was statistically significantly different. So we wanted to explore why this was happening. And in this image, you can see how, in this case, both paws were reduced in the number of touches. And we were thinking about the explanation for this because we were expecting the use of one limb or the other. And we think that once the neurons are completely integrated into the circuitry of this brain, when you shut down this area of the brain, is not only affecting this side, but also the other one, because both hemispheres are connected through the synapses. So we start thinking that the silencing of one area of the brain is not only affecting this side, but also both. And to try to explain or to demonstrate this hypothesis, we did a control experiment where we were injecting the allorodopsin in both sides of the cortex, in both hemispheres, and we were illuminating one or the other to try to reduce, to silence the activity of these sides of the cortex. And we observe exactly the same result as we observe in our transplanted animals, that when we were sitting down one side or the other, both legs were affected in the same way. So there was not a preferential use of one side, but the amount of touches of both paws was reduced in the similar way. So we demonstrate that when we transplant the cells, these cells are contributing to the functional recovery in the same way as it's happening in the normal intact brain without any damage. So to go further, in the last year, we also published a paper developed in Stem Cell Center in Lund, in the laboratory of Salko Kaya, where we wanted to explore what will happen if we use tissue that is human origin as a recipient for the transplant. Because what I was showing during all this talk is related with the transplantation of human cells into an animal. But we don't know what will happen if we transplant these cells into the human tissue. So to try to explore this possibility or this mechanism, we used pieces of brain that were donated to the laboratory from surgeries to generate organotypic cultures of human brain cortex. And we use this as the host for the transplantation of the LTNS cells. And this work was done by 3 postdocs in Salko Kaya's lab, Marita, Cecilia, and Sara Palma. And they were exploring this kind of model to try to see if the cells can be connected in the same way as we observe in the animals. And here you can see images of these kind of cultures with organotypic cultures with the transplant. And in the image below, you can see how using electron microscopy, we were able to detect these synapses form in both directions, from the transplant to the slide and from the slide to the transplant. Also using the Ravious virus technology, in the same way as before, we were able to demonstrate the connections from the slide to the grafted cells, and in the other way around, from the grafted cell to the transplant, to the slide, the cortical slide from a human patient. So to go one step further, we wanted also to see if these cells were receiving some kind of information, and to do that, in these slides, we record with electrophysiological recordings in the grafted cells to try to see if they were receiving information, and we were able to detect this spontaneous activity arriving to the transplanted cell. With this set of experiments, this paper was published, as I said, last year in Stem Cell Translational Medicine. We demonstrate that this integration of the cells, transplanted cells, and the host tissue is happening also in a human-to-human device or setup. So to end up with my talk today, I just want to comment a bit about my future plans and what's going on now in my laboratory in the future and in the future projects. In this case, what I'm gonna try to incorporate is a new tool that we have in the lab, that is this GCAM molecule, or GECI. Those are genetically encoded calcium indicators that we can use to follow in vivo the activity of the neurons, because the calcium levels that we can follow with these kind of molecules are changing with the activity of the neurons. And in this kind of experiments, I have very valuable help from Jordi Soriano, one of our collaborators, in this case in the physics faculty of the University of Barcelona, that is developing different tools to analyze how the networks are maturing or are integrating during the process of generation of the network or integration of transplants online using this kind of tools. So in this, using this tool, we are now trying to explore how the LTNS cells or the human progenitors can generate networks that are connected one to each other. So the neurons are able to generate a circuit that can be connected as a whole. And then in these images, in this video, I'm showing you how the activity of the calcium imaging is in this kind of cultures when they are 21 days after starting the differentiation. This is a very immature culture where we can see very random activity of the neurons without any connectivity between the neurons. But if we wait for some time, this is a video of 32 days, we can observe how some of the activities are becoming more synchronous. So all the cells simultaneously are firing together in synchronous events. And this is what is telling us about when the cells are connecting one to each other. So if I'm showing the last time point at 48 days of culture, we can see how in this case, most of the neurons will fire together in all the events being a synchronous activity. So this is telling us about how the culture is getting more mature, and we can use this in our purpose, in our model, to try to explore what is happening in our brains after transplantation. So to do that, we were designing in our laboratory, this work was done by Clelia Introna, one of the PhD students in the laboratory, in the histology unit. She was designing a lentiviral plasmid to incorporate the GCAM with a puromycin selection cassette. So we are able to generate a cell line that is expressing in a stable way the GCAM in all the cells. And this is under the promoter of synapsin. So under synapsin promoter, so only the neurons will express this GCAM. So we have incorporated these particles into lentivirus, and together with the rest of the people in my lab last year and also right now, we are now using this plasmid or this lentivirus to modify the LTNS cells before transplanting. And our idea is to use these cells in the model of ischemic stroke to try to see online in the animal how these cells are connected and how is the dynamics of this integration, functional integration of the cells into the brain. So with this, I just want to acknowledge all the people in my lab and all the institutions that I belong to, also the rest of the people of the histology unit in the Faculty of Medicine in the University of Barcelona, also the agencies that are trusting on us, giving us money to do our projects. Importantly, I want to thank all the people in Sal Kokaya's lab, where most of the work that I have been presenting was done, especially in the previous, in the first stages of these projects. Also, I would like to thank Malin Parmar and Shane, because they were sharing with us the Ravious virus technology. Galina Skibo and Oleg were doing all the electron microscopy, and Karl Diserov was helping a lot with the optogenetic tools. And thank you all for listening. I'm open to get questions at this point.
All right. Thank you very much, Daniel, for this very interesting talk. So the paper is now open for discussion. So if you want to make any questions, please use the chat option that you have in the bottom of your screen, and just state your name, and I will give the floor in a first come, first serve basis. So in the meantime that we receive the first question, let me just remind everyone that this webinar series is now open to everyone, not only to CIBERNED researchers, but everyone outside CIBERNED, both nationally and internationally. The webinars are held usually every other week always on Thursdays at 3:30 p.m. And you can check the program for the next 6 months in the CIBERNED webpage and also through the social networks from CIBERNED as well. So I haven't received any request for questions yet. So, but please don't hesitate to send me a short message with your name through the chat option so I can give you the floor and you can make the questions directly to Daniel. Yeah, we have the first question from Awa Fuster. So please just make sure that your microphone is on, and if you want to also turn your camera on, that's fine. I think it will be easier for Daniel to see who is posting the questions. So just go ahead.
Yeah, hello. Well, first of all, thank you for your talk. And then my question was, how far do you think we are of applying these therapies you were explaining in real clinical cases?
Okay, thank you, Alba, for the question. Well, at some point, this is a common question that we are getting when we explain our work. I'm not the only one working in this field, and this most of this research was done using a stroke model, but in many other neurodegenerative diseases, this technology can be in the same way used. And there are other studies, for example, using Parkinson as a model, where all this technology is very close now to be in the clinic. There are many clinical trials using patients that are now ongoing. These transplantations with stem cells in the patients started a long time ago. Importantly, also, I think it's important to highlight that in Lund, there were pioneers like Ole Lindvall doing transplantations in these kind of patients, Parkinsonian patients using embryonic stem cells, and the results were quite important. And now we are concluding a lot of things about this project. So now all this field is exploding in Parkinson's disease. I think very close in 1 or 2 years, we will see results very interesting with patients where we can see a huge recovery of the patients with a lot of data to be analyzed. And I think in the same way in the stroke will happen. We still have to be sure about what's the best protocol to transplant the cells. We need to know exactly, for example, how many cells and when it should be transplanted into the patient, because we know that there is a critical window where we can transplant the cells. We cannot transplant the cells a few months later after the stroke, but during the first month, the plasticity in the damaged area is still enough to get good results, success with the transplantation. So all these things should be more or less established, and we have to put a lot of emphasis in how we develop this technology or these protocols. But also in parallel, there are many works that has been doing, is doing now in the world, for example, in Japan and US, trying to generate bank of cells with a lot of frozen vials of human cells that we can transplant into this kind of patients. This, for example, the work that Lawrence Studer is doing in US or Jun Takahashi in Japan, is they are trying to generate a whole bank of cells that we can take to transplant once we need the cells to do this kind of regenerative medicine. And this is also a nice and important work that should be done in order to do this therapy more reproducible and more efficient and successful. And those are many different fields or lines that are in parallel going to the close application of this kind of therapies in this kind of patient. But I think in stroke patients, There are also some clinical trials, very a few of them with a few patients, but with the time, with a few years, we will see how this number is increasing, and probably in 5 or 10 years, we will see the results of this.
Okay, thank you so much.
Thank you.
All right, thank you both. Again, if anybody else is interested in making a questions, just please so by using the chat option and send just a message stating your name. I haven't received any other requests yet. Let's wait for a couple of minutes. No questions? All right. If there are no more questions, then we're going to close the session here. Not before thanking Daniel for his time and his willingness to share his work with all of us. And thank you everyone for attending, and remind you that we'll have another webinar in 2 weeks on Thursday at 3:30 p.m. As always. So thank you very much everyone for attending, and thank you Daniel once again, and I'll see you hopefully in the next webinar. Have a good day. Bye-bye.